All right. Hello, everyone. Thank you so much for giving me the opportunity to talk about my postdoc project with you today. So in the Gomez group, our research focuses on glioblastoma. So glioblastoma is one of the most common and aggressive forms of primary brain cancer, and although it's quite rare, it has disproportionately high morbidity and mortality. So to give you an overview, when a patient presents with glioblastoma, the current standard of care combines both surgical resection followed by chemotherapy and radiotherapy. However, tragically, tumour recurrence is inevitable as therapy-resistant tumour cells eventually repopulate the brain. So as such, this disease has a really, really poor prognosis with 5-year survival rates of less than 15%, and this has not improved in the past 15 years, so we need to do better. So why is this the case? More and more research has shown that it's actually the tumour microenvironment that can contribute to therapy resistance. So the tumour microenvironment is the tissue context in which tumour cells reside. And the tumour microenvironment is quite complex. It contains many different kinds of tumour subpopulations that are distinct in terms of their gene expression, but also many non-malignant populations. So the normal brain cells that you have resident in your brain are also part of the tumour. So this includes immune cells and blood vessel cells and neurons and oligodendrocytes. And this can vary from patient to patient. And it is thought that it is this complex microenvironment that actually supports the tumour cell growth by forming these little supportive niches in the brain. So if we want to investigate new therapeutic avenues, one avenue we can look at is to disrupt these microenvironmental interactions between tumour cells and surrounding cells. But this would require a greater understanding of the supportive networks that exist within the tumour microenvironment and a more patient-specific view of the disease. And one area that we're particularly interested in looking at is tumour metabolism. So why tumour metabolism? So what makes cancer cells quite distinct is that they have a really, really high energy and metabolic demand because they grow a lot and they divide a lot. So they need a lot of fuel sources to be able to do that. So it's thought if we can therapeutically target cancer metabolism, we might be able to drive cancer cells towards senescence and death. But tumour metabolism is not so straightforward. So tumour cells can get their energy and their resources from many different kinds of fuel sources. And importantly, even if a tumour cell is quite reliant on one fuel source, if that tumour cell becomes deprived of it, it can undergo metabolic reprogramming. That is, it can change and pivot its metabolism to utilise alternative fuel sources to continue to survive under stress. And we hypothesise that it's actually the microenvironment that can help tumour cells do this metabolic reprogramming. So if a tumour cell becomes deprived of a fuel source that it needs, it can signal to its microenvironment, which can induce metabolic reprogramming around the surrounding cells around it, so that those cells supply the metabolites and resources that it needs to continue to grow and divide. And importantly, we hypothesise that this is actually going to be quite varied across the tumour landscape and as well between patients because there's different environmental constraints across the tumour. So my project is to build a map of the tumour tissue landscape. And to do this, I need to answer 3 questions. So what are the metabolic pathways that are active in different populations within the tumour? What do the cell communities look like? So what cells reside close to each other that they could be having these metabolic interactions? And what is the spatial distribution of those fuel sources, of those metabolites across the tumour? And hopefully with all this information, I can build that map. So to answer that first question, I'm employing a technique that's called single-cell RNA sequencing. So this is a technique where we can take patient tissue samples, their tumour samples, dissociate them into all the individual cells that make up that tumour, so the malignant populations as well as the healthy normal cells that reside there. And then we can then genetically sequence each individual cell to see what its gene expression looks like. And so far we've now done this for, we've included data for 56 patients, so quite large data sets that crash my computer a lot and hopefully we'll upgrade soon. But we can see very beautifully that these cells have got different metabolic pathways active. So there's 2 heat maps here. Those pathways that are active are in red, those pathways that are a bit lower in activity are blue. And we can see different malignant tumour populations have got different metabolic pathways active. But this is also true for those non-malignant brain cells as well. They have got different metabolic pathways active. So now we need to see where they reside in space. Are they residing in close proximity to each other to help those metabolic interactions? So to do this, I'm then employing a technique that's quite new that's called spatial transcriptomics. This is a really exciting technique where we can take patients' brain tumour tissue slices, mount them onto these specialised slides that have got these barcoded spots. So these barcoded spots allow the gene transcripts to be bound to special oligos that once you then undergo sequencing, you can trace where that gene transcript came from within the tissue and build a map of gene expression across the whole tissue. And that allows you to see the composition of different cells and pathways that are active. So this is some of our data. You can see a patient's tissue slice from their brain tumour, and we can see these different tumour malignant populations. So here you've got oligodendrocyte-like tumour cells and mesenchymal-like tumour cells that reside in very different locations in the tumour landscape. We can also look at metabolic pathways. So OXPHOS and glycolysis are two classic energy-producing metabolic pathways that have got different fuel source requirements. And we can see that our two different populations of tumour cells here reside in very different areas of the brain that have got different fuel source requirements. And we can also look at non-malignant cell populations. So here we've got immune cells, and here we've got blood vessels. And we can see, for example, that these oligodendrocyte-like tumour cells reside in landscapes which are high in immune cell populations, whereas our mesenchymal-like tumour cells reside in landscapes that are low in immune cells. So we can start to build that map of what the landscape looks like in terms of cell composition and metabolic pathway activity. But we still have got a missing piece, which is the metabolites themselves. So there's another technique that I'm employing called mass spectrometry imaging. So this technique also allows us to take patient tumour slices, mount them onto slides, and then fire a laser at that tumour, pixel by pixel, and look at what the spectral profile of metabolites are so that we can then map where those metabolites are across the tumour landscape. So this part of my project is still in progress, but so far we've generated some mass spec imaging data for both small metabolites like sugars as well as large metabolites like fats and lipids from 8 glioblastoma samples from 7 patients, and our aim is to increase that number so we can look at the diversity across multiple patients. But we can see here, for example, we've got 2 different metabolites that reside in very different spatial niches within the tumour. And the next step is then to assess the distribution across those different tumour regions. And hopefully we can then combine all this information together to be able to create that map of the tumour landscape in terms of its metabolism. And that will allow us to then pinpoint those pathways that look the most interesting and the most important for tumour cells to survive. And we can then do some functional testing in our in vitro models. So in the Gomez group, we use patient-matched glioblastoma organoids. These are produced by taking patients' resected tumour tissue, chopping them up into little pieces, and then growing them in culture long-term. And those matched glioblastoma organoids are going to be comparable to their patient's tumour in terms of gene expression, as well as what the metabolic landscape and the cell composition looks like of those parent tumours. So we can then do drug studies and knock down genes in metabolism pathways that we think are interesting and see if any of those contribute to preventing growth and inducing death in tumour cells, which we can then hopefully take towards clinical trial. So with that, I would obviously like to thank many people. This is a collaborative project with many people putting a lot of energy in. So obviously I'd like to thank the Gomez group itself, so especially Guillermo Gomez, who gave me the opportunity to work on this project, as well as I'd like to thank Dr. Ashwini Patil. She is the CEO of a company in Japan which does a lot of bioinformatics work, and she's helped us do some work on these big patient data sets. And I'd also like to thank as well all the clinicians and patients that donate their tumour tissue to help us, and that's what makes it possible to do projects like this. And lastly, I'd like to thank all our funding partners, including the NRF, for giving us the opportunity to explore this project. So thank you very much for your time.
Stay up there, Chloe.
Okay.
I might just, we debated whether we would have some questions here. I think I'd like to have some questions. If anybody's got any questions about this, I can start it off maybe while people are thinking. You don't have to ask questions, but it's good because this talk is probably more at the fundamental side of things, of understanding fundamental biology around brain tumours. And we'll progress sort of through other aspects of brain tumour research during this day. So how long do you think, obviously you've got some pathways you're looking at, and this is incredibly cutting edge stuff. Nobody else that I know of in the world is really combining this type of cutting edge technologies in brain tumour space. So how long do you think, if you identify a pathway, how long do you think it will be to be able to initiate that clinical trials?
Well, it's a hard question, but we've got some pathway candidates that we're already interested in. So glutamate metabolism pathway is one. We already know from some literature that if you deprive cancer cells of some resources, they become really dependent on glutamate to continue to grow. So there are some really great candidates that we can see also coming up in our data. We can then do some functional testing. Hopefully that would take us about a year and a half or so to get some good data. And we do have some collaborations with clinicians based in Melbourne that hopefully we can reach out to start to see what their interest is, if it would work for a clinical trial. And so I hope within the next 5 years that we might be in that space to be able to take it forward.
And I guess that's the other thing that you need to be aware of. If the audience is aware of this, the Brae's Comprehensive Cancer Centre, the building is going up two doors down. It's a clinical trials focused building. So the idea is that, what I was just saying, that there's, clinicians in Melbourne, there are great clinicians here and Adelaide as well. The clinical trials have not necessarily always been at the front of mind. I think this BCCC building that's coming up will be actually really bringing that to the fore in Adelaide and allowing clinical trials to run out of Adelaide.
Which would be fantastic, yeah.
Perfectly dovetail into that. So.
Thank you.
Any other questions? There's be plenty of time afterwards. People are often a bit scared about asking questions in the forum, but there's plenty of time to talk. Yes?
Thank you. Look, as a past fundraiser and working in philanthropy, and someone who's diagnosed with GBM, but it's not there at the moment, total radical resection. My big question to you is, for you to do this project and continue to do what you do, how much do you need per annum to fulfil your mission? Our mission.
Oh, it's a really good question.
And then add 20%.
For a lot of, so especially the spatial transcriptomics is quite an expensive experiment to do. So it takes about trying to remember exactly, about $10,000 or so to be able to do one sample. And obviously we want to increase that by a lot. So that gives you, I guess, an estimate of some of these experiments are quite costly. And as well, we need to update just our computing power, because we're now working with data sets which are 56 patients big, if not more. We want to include more into that, and each of those data sets is hundreds of thousands of cells, so, and all their gene transcripts. So we need to update our computing power to be able to do that. So yeah, we're looking at investing in a few things hopefully soon and reaching out for some funding to help us be able to continue what we do.
I think with this sort of project, the answer is almost unlimited, because Guillermo got a $2 million grant a couple of years ago. I think that's $500,000 a year, so over a 4-year period. But really, that's sort of scratching the surface, really. And I think with this sort of project, the more money, the more progress we can make in a faster period of time, right? So it's that's the simple answer.
Well, that and just increasing the number of samples. Glioblastoma is so diverse across patients. Each patient's tumour looks quite different from each other patient's tumour. There are probably similarities between them, but if we want to capture that diversity, we need a large number of samples to do that. So that's a limiting factor. We can do small scale projects, or we can do big scale projects, which would mean a lot more.
All right. Another question about that?
What's feeding the cells? So the most common metabolite that's utilised by a lot of cancer cells is glucose, so sugars. But as I was saying, they can pivot to use a lot of different kinds of fuel sources. So they can also use some fats as well, and they can use some amino acids. So amino acids come from proteins. And the idea is that it is surrounding cells in the brain that might be contributing to that metabolite pool. So whether it's even some of the neurons, so they almost feed off of the normal metabolism that's happening in your brain and take advantage of it, because your brain uses a lot of resources. It's one of the highest energy tissues in the brain, in the body. So they kind of take advantage and almost parasitically take advantage of their surrounding tissue. So yeah.